Welcome to the deep dive. Today we're taking on a truly fascinating aspect of your body's chemistry. We often think of amino acids as simply the building blocks for proteins, right?
That's the common view, absolutely.
But that's just one part of their story. We're going to dive into their critical, sometimes surprising role in generating energy and how your body brilliantly manages their breakdown.
Right, it's a journey into, well, intricate molecular mechanisms, biochemical pathways, and how it all integrates into your metabolic system.
Exactly.
And our deep dive today is directly inspired by the foundational insights found in Chapter 18 of Lehninger Principles of Biochemistry.
Ah, Lehninger, good source.
Indeed. It's all about understanding that intricate dance of molecules that allows your body to extract energy from amino acids and safely deal with the byproducts, especially that nitrogen.
And our mission for you is to unpack precisely how your body handles those nitrogen-containing amino groups to prevent toxicity, and then cleverly repurposes the remaining carbon skeletons for energy or other vital molecules.
Consider it your shortcut to understanding this complex biochemical landscape.
Yeah, mixing essential knowledge with some truly aha moments that we think will keep you hooked.
Hopefully.
So, let's set the scene. Think about where amino acids come from. Your diet, of course, but also the constant turnover of proteins within your own body.
Right, that internal recycling is significant.
They're a major energy source, especially for carnivores or, during specific metabolic conditions like starvation or uncontrolled diabetes.
When glucose might be scarce or unusable.
Exactly. The key insight here is that when amino acids are used for energy, they effectively split into two main parts. Their amino groups, with the nitrogen, and their carbon skeletons.
And each part takes a distinct and critical journey.
Okay, so where does that journey begin? Digestion.
Precisely. It starts right in your digestive tract. When you eat protein, it first hits your stomach. The acidic environment, thanks to hydrochloric acid, does two things. It acts as an antiseptic, killing off unwanted bacteria, and it starts to unfold or denature those complex proteins, making them easier to break down.
Makes sense. Easier targets for enzymes.
Exactly. And your stomach also releases an inactive enzyme precursor called pepsinogen.
Inactive, so it needs activating.
Correct. That low pH in your stomach triggers pepsinogen to essentially activate itself into pepsin.
Clever.
And pepsin then starts chopping the long protein chains into smaller peptides. Just the initial cuts, really.
Right. Then it moves to the small intestine.
Yes. As those partially digested proteins move into your small intestine, your body kicks off another phase. Hormones like secretin tell your pancreas to release bicarbonate.
To neutralize the acid?
Precisely. Then another hormone, cholecystokinin, triggers the pancreas to release a whole suite of other inactive protein-digesting enzymes, zymogens like trypsinogen and chymotrypsinogen.
More inactive precursors.
Loads of them. And here's the clever part. An enzyme from your intestinal cells called enteropeptidase activates trypsinogen into active trypsin.
So one key activates the main one.
And once active trypsin is present, it creates cascade, activating all the other pancreatic enzymes. It's like a master switch.
This elaborate dance of inactive precursors, or zymogens, becoming active only later, that strikes me as incredibly clever. Why the complexity?
It's a brilliant safety switch. Imagine these powerful protein-digesting enzymes activating too soon inside the very cells that make them, especially in your pancreas.
Oh, that would be bad. Very bad.
It would be devastating. Self-digestion, basically.
Mm-hmm.
And sadly, when this system does fail, as we see in acute pancreatitis,
Right, I've heard of that.
those enzymes activate prematurely, causing immense pain and organ damage. It can actually be fatal.
Wow, so the zymogen strategy is critical.
Absolutely vital. But when it works, these enzymes efficiently break down proteins into individual amino acids.
And those amino acids, where do they go?
They get absorbed through the intestinal lining into your bloodstream and head straight to the liver. The liver is the main processing hub.
Okay, liver central. So what's the first thing the liver does with them?
Once those amino acids arrive, the pivotal first step in using them for energy is removing their amino groups, getting that nitrogen off.
Right, how?
This happens through a process called transamination, primarily catalyzed by enzymes called aminotransferases, or sometimes called transaminases.
Okay, aminotransferases.
These enzymes act like shuttles. They transfer the amino group from an amino acid onto a key molecule called acetoglutarate.
Ketoglutarate. That sounds familiar. Citric acid cycle.
Exactly, a direct link. So the amino group goes on to acetoglutarate, forming L-glutamate and leaving behind an oxido acid version of the original amino acid.
So it's not getting rid of the nitrogen yet, just moving it.
Precisely.
Yeah.
There's no net deamination at this stage. Think of it as a central collection point. Amino groups from various amino acids are all funneled into glutamate.
Making glutamate the main collector.
Yes, and a crucial helper in all these aminotransferase reactions is pyridoxal phosphate, or PLP.
PLP, vitamin B6 derivative.
That's the one. Derived from vitamin B6, it acts like a temporary chemical hand or carrier. It picks up the amino group from one molecule and then delivers it to ipidoglutarate.
Like a little intermediate holder.
Exactly. Its ability to temporarily carry that amino group is absolutely essential for these transamination reactions to occur.
Okay, so we've collected all this nitrogen, mainly in the form of glutamate, in the liver. Now the critical question, how does your body manage that collected ammonia to prevent its inherent toxicity? Because free ammonia is bad news, right?
Extremely bad news. So in your liver cells, specifically hepatocytes, the glutamate we just collected is transported into the mitochondria.
Inside the mitochondria, okay.
There, an enzyme called L-glutamate dehydrogenase catalyzes what's called oxidative deamination.
Oxidative deamination. Sounds like it removes the amino group and does some oxidation.
That's exactly right. It removes the amino group as an ammonium ion, NH4+ sort, regenerating the ipidoglutarate we used earlier. And it also transfers electrons, usually to NAD+ or NADP+R.
Interesting, it can use both NAD+ and NADP+.
It's quite unusual. And what's also remarkable is this enzyme's activity is allosterically regulated.
Meaning it can be turned up or down?
Yes. ADP, which signals low energy in the cell, activates it. GTP, signaling high energy, inhibits it.
So it's linked directly to the cell's energy status, like a dimmer switch.
A perfect analogy. It allows your body to tune nitrogen processing based on energy needs.
And here's a surprising fact you mentioned earlier, linking this enzyme to a specific condition.
Yes. There's a genetic disorder called hyperinsulinism, hyperammonemia syndrome. It's caused by a mutation that essentially locks glutamate dehydrogenase in the on position.
Permanently active. What happens?
It leads to both high blood ammonia levels, because it's constantly releasing ammonia, and also problems with insulin regulation leading to low blood sugar. It vividly shows how this single enzyme connects carbon and nitrogen metabolism.
Wow, okay, so that's in the liver. But other tissues produce ammonia too, right? How does it get to the liver safely?
Excellent question. Given ammonia's high toxicity, especially to the brain, its safe transport is critical. In many tissues outside the liver extrahepatic tissues, any free ammonia generated is quickly mopped up.
No.
It's combined with glutamate to form glutamine. This reaction is carried out by an enzyme called glutamine synthetase, and it requires energy from ATP.
So glutamine is a safe transport form.
Exactly. Glutamine is non-toxic and can travel safely in your bloodstream to the liver, or, interestingly, the kidneys.
Kidneys too, why there?
Well, once in the liver, or kidneys, glutamine is converted back to glutamate and NH4+ by an enzyme called glutaminase.
Releasing the ammonia again?
Yes, but now in a controlled place. And there's a fascinating real-world connection here. In conditions like metabolic acidosis, where your blood is too acidic.
Okay.
Your kidneys actually ramp up their glutamine processing.
Yeah.
They release the NH4+ directly into the urine. This helps get rid of acid and simultaneously generates bicarbonate to help buffer the blood.
That's clever. Using nitrogen disposal to manage acid-base balance.
Very clever. And there's another crucial ammonia shuttle, particularly important for your muscles.
Muscles, what's special there?
It involves the amino acid alanine via the glucose-alanine cycle. When your muscles are working hard, they break down proteins for fuel, producing ammonia. They also produce lots of pyruvate from glucose breakdown.
Right, lots of metabolic activity.
So rather than releasing toxic ammonia, the muscle cleverly combines the amino group with pyruvate to form alanine.
Alanine becomes the transport molecule from muscle.
Correct. Alanine travels safely in the blood to the liver. In the liver, alanine is converted back to pyruvate, which the liver can use to make new glucose to send back to the muscle.
Ah, closing the loop.
And the amino group goes on to glutamate, releasing the ammonia safely within the liver for disposal. This cycle effectively shifts the energetic burden of glucose synthesis and urea production to the liver.
Letting the muscles focus on contracting, that's efficient.
Very efficient resource management.
So what does this all mean for you, the listener? It really hammers home that ammonia toxicity is a serious problem. Ammonia is incredibly harmful, especially to your brain.
Absolutely. High levels can lead to severe neurological effects, cognitive impairment, loss of coordination, tremors, slurring of speech.
Even seizures.
Yes, seizures too. And in the most extreme acute cases, it can cause fatal brain swelling or cerebral edema.
Why is it so bad for the brain? What's the molecular basis?
It's quite intricate. Ammonia, specifically the uncharged form NH3, can slip across the blood-brain barrier quite easily.
Sneaks right in.
Pretty much. Once inside brain cells, it gets protonated to NH4+ and accumulates.
Yeah.
Your brain's support cells, the astrocytes, try desperately to detoxify it by converting it and glutamate into glutamine using that glutamine synthetase enzyme we mentioned.
But that causes problems.
Two main problems. First, it can deplete the astrocyte supply of glutamate, which is not only needed for this reaction, but is also a precursor for GABA, an important neurotransmitter. So neurotransmitter balance gets disrupted. Second, the high levels of glutamine inside the astrocytes create an osmotic imbalance, causing them to swell up. And the NH4+ itself can interfere with potassium channels and transporters, disrupting normal ion balance and neuronal function.
Leading to astrocyte swelling and neuronal overactivity, explaining the seizures and incoordination.
Exactly. If it persists, that swelling, the edema, can become fatal.
Right. So clearly, we need a disposal system. Now that we've highlighted just how dangerous ammonia is, let's talk about your body's elegant solution for its disposal in mammals, the urea cycle.
Ah, yes, the urea cycle, a truly remarkable metabolic pathway, discovered in part by Hans Krebs, the same Krebs of the citric acid cycle fame.
Busy man.
Indeed. This process happens almost exclusively in your liver. It's the ingenious system that converts that toxic ammonia plus another nitrogen from aspartate into harmless, water-soluble, excretable urea.
A metabolic masterpiece, as you called it. How does it work? Step by step.
It involved 5 enzymatic steps, starting in the mitochondria and finishing in the cytosol.
Meto and cytosol. Okay. Step 1.
Step 1. Mitochondrial. The first nitrogen input, that free NH4+ released from glutamate, combines with bicarbonate, derived from CO2, to form carbamoyl phosphate. This reaction requires 2 ATP molecules and is catalyzed by carbamoyl phosphate synthetase I, or CPSI. This is the committed and major regulated step.
2 ATPs just to start. Okay. Step 2.
Step 2, mitochondrial. Carbamoyl phosphate then donates its carbamoyl group to a molecule called ornithine, forming citrulline. This is catalyzed by ornithine transcarbamylase. Think of ornithine as a carrier molecule that gets regenerated at the end, a bit like oxaloacetate in the citric acid cycle.
So ornithine carries the group.
It accepts the carbamoyl group.
Right.
Then citrulline moves out of the mitochondria and into the cytosol.
Out to the main cell area. What happens there?
Step 3, cytosolic. Here comes the second nitrogen atom. Citrulline condenses with the amino acid aspartate to form arginosuccinate. This step requires another ATP, effectively costing 2 high-energy bonds, and is catalyzed by arginosulfonate synthetase.
So aspartate brings in the second nitrogen for urea.
Precisely. Step 4, cytosolic. Arginosuccinate is then cleaved by arginosuccinase into arginine and fumarate. Interestingly, this is the only reversible step in the cycle.
Fumarate. Another citric acid cycle link?
Absolutely. We'll come back to that. Step 5, cytosolic. Finally, the enzyme arginase cleaves arginine to yield urea, that's our final product, and importantly, it regenerates ornithine.
And the ornithine goes back to the mitochondria.
Back into the mitochondrion to pick up another carbamoyl phosphate ready for another round.
You know, it's not just a series of isolated steps. You mentioned earlier this idea of metabolons. How does that apply here?
Right. There's evidence suggesting that several of these urea cycle enzymes, particularly the cytosolic ones, are physically clustered together. They form a sort of supramolecular complex, a metabolon.
Like a little assembly line.
Exactly. The idea is that the product of one enzyme is passed directly, or channeled, to the active site of the next enzyme in the pathway.
Increasing efficiency.
Tremendously. It prevents intermediates from diffusing away or participating in side reactions. It really highlights the remarkable organization within the cell.
And you mentioned the links to the citric acid cycle, fumarate and aspartate.
Yes, this is often called the Krebs bicycle, a really elegant metabolic interconnection. The fumarate, generated in step 4 of the urea cycle, can be converted to malate in the cytosol.
Okay.
Malate can then enter the mitochondria and join the citric acid cycle, eventually being oxidized back to oxaloacetate, generating energy in the form of NADH.
So the urea cycle feeds into energy production.
It does, and the connection goes the other way too. Aspartate, which brings in that second nitrogen atom in step 3, is typically formed in the mitochondria from citric acid cycle intermediates like oxaloacetate, via transamination.
Ah, using glutamate again.
Often, yes.
Yeah.
Then this aspartate is transported out to the cytosol to participate in the urea cycle. There are specific transporters, like the malate aspartate shuttle, that facilitate this exchange and also help move reducing equivalents in ADH between compartments. It's deeply intertwined.
That interconnection is fascinating and clinically relevant.
Very much so. Remember those aminotransferases we talked about earlier, involved in collecting amino groups onto glutamate? Two key ones are alanine aminotransferase, ALT, and aspartate aminotransferase, AST.
Right, ALT and AST, liver function tests.
Exactly. These enzymes are normally contained within liver cells. But if the liver is damaged by infection like hepatitis or toxins like alcohol or other diseases, the cells can lyse or leak.
And the enzymes spill out.
They spill out into the bloodstream. So measuring elevated levels of ALT and AST in your blood serum is a crucial diagnostic indicator of liver damage. It directly reflects the health of these metabolic pathways.
So how does your body regulate such a vital, potentially dangerous if malfunctioning process like the urea cycle?
Regulation occurs at two main levels, long-term and short-term.
Okay, long-term.
Over the long term, the actual synthesis rates of the 5 urea cycle enzymes plus CPSI adapt to metabolic needs. For instance, if you switch to a very high-protein diet or during prolonged starvation when body protein is broken down for energy.
More nitrogen needs processing.
Right. Your liver responds by synthesizing more of these enzymes over hours or days to increase its capacity for urea production. Conversely, on a low-protein diet, the synthesis rates decrease.
Makes sense. And short-term, like minute to minute.
On a shorter time scale, the key regulatory point is that very first enzyme in the mitochondria, carbamoyl phosphate synthetase I, CPSI.
The one using the first ammonia and 2 ATPs.
That's the one. It's allosterically activated by a molecule called N-acetylglutamate.
N-acetylglutamate, where does that come from?
It's synthesized from glutamate and acetyl-CoA by another enzyme, N-acetylglutamate synthase. And the activity of this synthase is stimulated by arginine.
Arginine, an intermediate in the urea cycle itself.
Yes. So when amino acid breakdown increases, levels of glutamate rise. If arginine levels are also high, indicating the cycle is active, N-acetylglutamate is synthesized.
Which then activates CPSI.
Exactly. It's a feed-forward activation signal saying, hey, we have lots of amino acids coming in. Let's ramp up the urea cycle right now. It ensures the cycle responds quickly to ammonia load.
Seems efficient. Now, what about the energy cost? It looked expensive, 4 high-energy bonds per urea.
It does seem costly. 3 ATP molecules are consumed, yielding 2 ADP and 1 AMP, which is equivalent to hydrolyzing 4 high-energy phosphate bonds. However,
there's a catch.
There's that Krebs bicycle connection. The fumarate produced enters the citric acid cycle via malate. The oxidation of malate to oxyl acetate generates 1 molecule of NADH in the mitochondrion.
And NADH means ATP later via oxidative phosphorylation.
Right. Each mitochondrial NADH yields about 2.5 ATP. So the energy generated from processing the fumarate significantly offsets the initial ATP cost of the urea cycle. The net cost is much lower than it first appears.
A clever metabolic economy, but what happens if this system doesn't work? Genetic defects.
Yes, genetic defects in any of the urea cycle enzymes are extremely serious medical conditions. They all lead to an inability to properly convert ammonia to urea.
Resulting in hyperammonemia.
Exactly. Ammonia builds up in the blood. Infants born with severe deficiencies can appear normal at birth, but within 24-48 hours as protein breakdown begins, they rapidly develop symptoms. Lethargy, vomiting, irritability, poor feeding, rapid breathing.
The signs of ammonia toxicity we discussed?
Precisely. And brain swelling. Without immediate and aggressive treatment to lower ammonia levels, these conditions can cause irreversible brain damage or can be fatal.
That's devastating. How are they treated?
Treatment is complex and lifelong for many. A key strategy is careful dietary management, restricting protein intake to reduce the ammonia load, but not so much that you cause a deficiency of essential amino acids needed for growth and repair. It's a very fine balance.
Tricky. What else can be done?
Doctors often administer specific aromatic acids like sodium benzoate or sodium phenylbutyrate.
That's what those help.
These compounds provide an alternative pathway for nitrogen excretion. Benzoate combines with glycine, and phenylbutyrate, after conversion, combines with glutamine. These conjugated products are then excreted in the urine, effectively removing nitrogen from the body.
So they bypass the defective urea cycle to some extent?
Yes, they provide an alternative route to get rid of waste nitrogen. More specific treatments depend on the exact enzyme defect.
Like?
For a deficiency in N-acetylglutamate synthase, the enzyme that makes the activator for CPSI, patients can be given an analog called carbamoyl glutamate. This directly activates CPSI, bypassing the need for N-acetylglutamate.
Clever workaround.
And in some other deficiencies, like ornithine transcarbamylase or arginosuccinase deficiency, simply supplementing the diet with high doses of arginine can sometimes help push the cycle forward or provide enough ornithine.
It really highlights how crucial each step is and this idea of essential versus conditionally essential amino acids.
Absolutely. Arginine is normally non-essential because we make it in the urea cycle. But if the cycle is blocked before arginine synthesis, it becomes essential, or at least conditionally essential.
Okay, we've covered the nitrogen journey thoroughly. Let's shift focus now. What about the rest of the molecule, the carbon skeletons of these amino acids?
Right. Once the amino group is removed, usually via transamination, you're left with an aikido acid, the carbon skeleton. The fascinating thing is that the breakdown pathways for the 20 different amino acid skeletons converge.
They don't all have unique paths.
No, they funnel into just a handful, 6 really major metabolic intermediates. These are molecules like pyruvate, acetyl-CoA, acetoacyl-CoA, acetylglutarate, succinyl-CoA, fumarate, and oxaloacetate.
Hey, those are all players in or connected to the citric acid cycle.
Exactly. That's the beauty of it. All roads lead to central metabolism. From there, these carbon skeletons can either be completely oxidized in the citric acid cycle to CO2 for energy,
Generating ATP.
or, depending on the body's needs and which intermediate they form, they can be diverted into making new glucose, that's gluconeogenesis.
If they form pyruvate or citric acid cycle intermediates other than acetyl-CoA.
Correct. Or they could be used to produce ketone bodies or fatty acids if they yield acetyl-CoA or acetoacyl-CoA.
It depends on the body's fuel status, essentially.
Precisely, which leads to classifying amino acids based on these fates.
Ah, the glucogenic and ketogenic distinction.
Right. Glucogenic amino acids are those whose carbon skeletons can be converted ultimately into pyruvate, or a citric acid cycle intermediate that can then be used to synthesize glucose via gluconeogenesis. Most amino acids fall into this category.
And ketogenic.
Ketogenic amino acids are those whose carbon skeletons are degraded into acetyl-CoA or acetoacyl-CoA. These cannot be used to make glucose, but they can be converted into ketone bodies or used for fatty acid synthesis.
Can they be both?
Yes. Several amino acids like tyrosine, phenylalanine, tryptophan, isoleucine, and threonine are both glucogenic and ketogenic because their breakdown yields different products.
And are any purely ketogenic?
Just 2, leucine and lysine. They are exclusively ketogenic. Their carbons cannot contribute to net glucose synthesis.
Okay, that's a useful classification. Now, breaking down these diverse skeletons must involve some complex chemistry. You mentioned PLP for transamination. What other cofactors are key players here?
Oh yes, several other crucial enzyme cofactors are involved, especially for reactions involving the transfer of single carbon atoms or one carbon groups.
One carbon transfers.
Yeah.
Sounds important.
It is, especially in amino acid and nucleotide metabolism. We're talking about biotin, which you might know from carboxylation reactions. It transfers carbon in its most oxidized state as CO2.
Right.
Then there's tetrahydrofolate, or H4 folate, derived from the B vitamin folate. This is incredibly versatile. It can carry one carbon groups in several different oxidation states, from methyl groups up to formal groups.
Folate, linked to development and anemia.
Critically linked. And then there's S-adenosylmethionine, or Adamet, often called SAM. This molecule is the major donor of methyl groups, the most reduced form of a one-carbon unit.
Biotin, tetrahydrofolate, Adamet, the one carbon crew.
Pretty much. And another important one, though not for one carbon transfer, is tetrahydrobiopterin, involved in some key hydroxylation reactions, like the one converting phenylalanine to tyrosine.
Okay. And you mentioned earlier a really interesting clinical link between B12 and folate.
Yes, this is a classic example of metabolic interconnection with huge clinical significance. Vitamin B12 is required for only 2 known reactions in humans. One is the conversion of methylmalonyl-CoA to succinyl-CoA, which we'll touch on later.
Okay.
The other reaction involves methionine synthase, which regenerates methionine from homocysteine. This reaction also requires folate, specifically in the N5-methyl tetrahydrofolate form.
So B12 and folate work together there.
They do. Now, if you have a vitamin B12 deficiency, often due to absorption problems leading to pernicious anemia, this methionine synthase reaction slows down or stops. As a result, folate gets trapped in that N5-methyl tetrahydrofolate form because that's the form used by the B12-dependent enzyme.
Trapped, meaning it can't be used for other things.
Exactly. It can't be converted back to other folate forms needed for synthesizing nucleotides, the building blocks of DNA and RNA. This impairment of DNA synthesis leads to megaloblastic anemia, where red blood cells and other rapidly dividing cells become large and immature.
Ah, so the anemia in B12 deficiency is actually due to a secondary folate deficiency?
Functionally, yes, it's the folate trap hypothesis.
Yep.
But B12 deficiency also causes serious, potentially irreversible neurological damage, likely related to the other B12-dependent reaction involving methylmalonyl-CoA.
And that's the danger.
That's the danger. If you treat someone with pernicious anemia, B12 deficiency, with high doses of folate alone, you can potentially correct the megaloblastic anemia, making the blood picture look better.
Huh.
But you haven't fixed the underlying B12 deficiency, so the neurological damage continues, perhaps masked because the anemia improved. It's crucial to diagnose the specific deficiency correctly.
A critical clinical point.
Yeah.
Okay, let's maybe look briefly at some examples of the breakdown pathways themselves. Which amino acids go where?
Sure, we can group them by their end products. For instance, several amino acids are degraded to pyruvate. These include alanine, just one transamination step away, serine, cysteine, glycine, threonine, and parts of tryptophan.
Glycine breakdown. Any clinical notes there?
Yes, a severe genetic defect in the enzyme complex that breaks down glycine, called the glycine cleavage system, leads to non-catotic hyperglycinemia. Glycine accumulates to toxic levels, especially in the brain, causing severe intellectual disability and seizures.
Grim.
Also, glycine can be converted to oxalate. While normally minor, excessive oxalate production can contribute to the formation of calcium oxalate kidney stones, a common and painful condition.
Ouch. Okay, what about amino acids ending up as acetyl-CoA or acetoacetyl-CoA, the ketogenic one?
Right, this group includes leucine and lysine, the exclusively ketogenic ones, plus isoleucine, threonine, tryptophan, and importantly, phenylalanine and tyrosine.
Ah, Phe and Tyr. Let's spotlight them. Their pathway leads to...
Their degradation pathway is quite complex, but it ultimately yields fumarate, which is glucogenic, and acetoacetyl-CoA, which is ketogenic. So they are both.
And defects here are well known.
Very well known. The most common is phenylketonuria, or PKU. This is caused by a genetic defect in the enzyme phenylalanine hydroxylase, which normally converts phenylalanine to tyrosine. Remember, tetrahydrobiopterin is the cofactor here.
So phenylalanine builds up.
Massively. You get shunted into an alternative pathway, forming phenylpyruvate and other derivatives. These give urine a characteristic mousy or musty odor. More importantly, the high phenylalanine levels are toxic to the developing brain, causing severe intellectual deficits if untreated.
But it's treatable.
Yes, thankfully. Newborn screening for PKU is standard in many countries. Treatment involves a lifelong, very strict diet low in phenylalanine. Enzyme replacement therapies are also emerging now.
A classic example of inherited metabolic disease. Any others in that pathway?
Briefly, alkaptonuria is another defect further down the Fatir breakdown pathway. It causes homogentisate to accumulate, leading to dark urine and later in life, arthritis, less severe than PKU generally.
Okay. What about pathways leading to acetoglutarate?
That pathway handles proline, arginine, histidine, glutamine, and glutamate itself. Glutamate can be directly converted via that glutamate dehydrogenase reaction we discussed. The others feed into glutamate first.
And succinyl-CoA, another citric acid cycle intermediate.
Yes, methionine, isoleucine, threonine, and valine feed into succinyl-CoA. The pathway involves that B12-dependent enzyme, methylmalonyl-CoA mutase.
Ah, the other B12 reaction.
Correct. A defect here causes methylmalonic acidemia, MMA, where methylmalonate and related compounds build up.
And you mentioned a compelling real-life case.
Yes, the tragic story of Patricia Stallings in the late 80s, early 90s. Her infant son tragically died, and she was wrongly accused of poisoning him. Later, her second child also became ill with similar symptoms.
Oh.
It was only through further biochemical investigation that it was discovered both children actually suffered from MMA, a genetic condition. The biochemical evidence eventually exonerated her. It's a powerful, albeit heartbreaking example of how crucial biochemical understanding is in medicine and justice.
Absolutely underscores the importance. Wow. Lastly, the branched-chain amino acids. They seem special.
They are somewhat unique. Valine, isoleucine, and leucine. Unlike most other amino acids, which are primarily catabolized in the liver, these three are mainly broken down in extrahepatic tissues, muscle, adipose tissue, kidney, and brain.
Why the difference?
Those tissues have higher levels of the initial aminotransferase specific for branched-chain amino acids. The liver lacks this enzyme. After the amino group is removed, the resulting keto acids are then oxidized by a large enzyme complex.
Let me guess, analogous to pyruvate dehydrogenase.
Very analogous. It's called the branched-chain keto acid dehydrogenase complex. It requires similar cofactors like thiamine pyrophosphate, lipomide, FAD, NAD plus AD, and CoA.
And a defect here causes.
A defect in this complex causes maple syrup urine disease, MSUD.
Aptly named.
Very. The accumulation of the branched-chain keto acids and the amino acids themselves imparts a characteristic maple syrup or burnt sugar odor to the urine and earwax. More seriously, it leads to severe neurological damage, feeding problems, coma, and can be fatal if untreated.
Treatment.
Like PKU, strict dietary control, limiting intake of valine, isoleucine, and leucine, often for life. Careful monitoring is essential. In some severe cases, liver transplantation can be curative because the liver does have some capacity for the later steps if it receives the keto acids.
Another stark reminder of the impact of single enzyme defect.
Absolutely. And finally, the simplest pathways. Asparagine and aspartate are readily converted to oxaloacetate, another key citric acid cycle intermediate. Asparaginase converts asparagine to aspartate, and then aspartate aminotransferase converts aspartate to oxaloacetate.
So to bring it all together then, the ultimate fate of these amino acid carbons is incredibly flexible. They feed into central metabolism.
Exactly. They can be completely oxidized for energy through the citric acid cycle and oxidative phosphorylation.
Or, if needed, diverted into making new glucose via gluconeogenesis, provided they yield the right precursors.
Or into producing ketone bodies, especially from leucine and lysine, or during conditions like starvation or uncontrolled diabetes when acetyl-CoA levels are high. It's all exquisitely tuned to your body's specific metabolic needs at any given moment.
A truly adaptable and dynamic system.
Absolutely.
Okay, let's try to unpack this a bit. What we've discussed today really highlights the incredible complexity, but also the efficiency of amino acid metabolism. It's quite something.
It truly is. From the intricate process of digestion and absorption
to the sophisticated mechanisms your body employs for safely handling and excreting nitrogen, like the urea cycle.
And then to the varied ways those carbon skeletons are dismantled and utilized, feeding into core energy pathways.
It's all deeply integrated into your body's overall metabolic network. You can't really separate it out. It's absolutely crucial for maintaining energy balance and, quite simply, for survival.
Couldn't agree more. And perhaps a final thought for you, the listener, to consider. Think about how even subtle defects, sometimes just a single non-functional enzyme in these specific, seemingly small molecular mechanisms can have such profound systemic impacts on human health.
Yeah, conditions like PKU, MSUD, the urea cycle disorders, MMA.
They really underscore the deep interconnectedness of biochemistry and clinical outcomes. They remind us that every pathway, every enzyme, plays a critical role in the delicate balance that is health.
A powerful connection indeed. Thank you for joining us for this deep dive into amino acid oxidation and urea production.
It was a pleasure.
We hope you gained a deeper understanding today and that this dive gives you plenty to mull over. Thank you for being part of the Last Minute Lecture family.